Hello friends, welcome again. This is chapter 8 in your CBSE Year 10 science book. How do organisms reproduce? So this is very important chapter, so understand this very well. Reproduction is asexual and sexual reproduction. So initially we'll deal with the asexual methods of reproduction and then move on to the sexual reproduction. So reproduction is the biological process by which new individual organisms that are called offsprings are produced from parent or parents. Okay, so it is a biological process in which new individual organism is produced from parent or organisms are produced from parents. It is a fundamental feature of all known lives. Each individual organism exists as the result of reproduction. If reproduction did not exist, none of us will be alive, neither plants nor animals nor human being. Okay? Now we've seen in one of the chapters, essential life processes, that nutrition, respiration, transportation, excretion, these are the essential life processes. But this list of essential life processes does not include reproduction. Okay, so reproduction is not essential to maintain a particular organism's life. So if, say, a human being is born, that human being does not need to reproduce to live a life. Okay, so it is not essential for that individual. So to maintain a life of individual, it is not essential, but it is essential to maintain the sustenance of the population of that individual. So if it is, so that's what is explained here. Why do organisms reproduce? If it is not essential to maintain the life, then why do they reproduce? So understand, when an individual organism reproduces, there is a lot of energy spent in this process. Then why should they reproduce, waste energy, waste their time for a process that is not even essential for their own survival? So let's try and understand that. So it is essentially a process by which individuals produce new individual of same kind. It is very important for existence of life on earth. Organisms reproduce to continue the chain of life, to pass on their genes which are acquired over millennia. Reproduction is necessary for the continuation of generation of particular species. Without reproduction, the species will extinct from the earth. It is also a source of genetic recombination, important step in evolutionary processes. So understand this, though it is not essential life process, it is necessary life process for sustenance or maintenance or continuation of generation of particular species. Okay, so that's the basic introduction, definition, and the need of reproduction. Now, next topic is, how do we know that two different individual organisms belong to same species? Now, it is easy if I ask you, how do you know two dogs belong to the same species? How do you identify dogs? You identify them because they look same, they look similar, they have the same body design, and that's how you say, oh, this is dog, that is dog, so they belong to the same species. So reproducing organisms, they create new individuals that look very much similar to themselves. Organisms look similar because their body designs are similar. Okay, so all the dogs will have a similar shape of the head, They will have 4 legs, a similar kind of tail, similar trunk. So that makes them look similar to their population of the same species. Now, do organisms actually create exact copies of themselves? Are they exactly same? So the answer is no. They are similar, they are not same. The organisms from same species look similar because their body designs are similar. Okay, now body designs are similar because the blueprints for these designs are similar. So reproduction makes the copies of these blueprints of body design. So blueprint decides body design, and because the body design is same, the organisms are produced that look similar to each other. Now, this is a simple explanation of how the copies of the blueprints are passed on from mother cell to the daughter cell. So these are the chromosomes. They replicate. So one chromosome gives rise to 2, one gives rise to 2. Then when the cell division starts, the replicated chromosomes separate. The cell creates 2 of its own kind. The chromosomes get distributed between these 2 newly formed cell structure or cell apparatus, and then there's a division between these 2 newly formed cells, and 2 daughter cells are formed. So you can see a copy of the blueprint, the DNA material is created. Okay, and this blueprint will create an organism with the same body design that will be similar to this, but it will not be exactly same. Why not? Let's have a look. The chromosomes in the nucleus of the cell contain information for inheritance of features from parents to the next generation in the form of DNA, that is deoxyribonucleic acid, and these are molecules. The DNA in the cell nucleus has the information for making proteins, and proteins are required to create new structures. Okay, so these DNAs, they have information. These are software. What information they carry? To create new proteins. And what do the proteins do? The proteins are used to make new bodies, new copies of these cells. Okay, so if the information is changed, if the information in the DNA is changed, different protein will be made, and change in the protein will lead to change in the body design. The creation of DNA copy is the basic event in reproduction. The cells use chemical reactions to generate their own DNA copies. So this DNA copying process is because of the chemical reactions. Okay, and chemical reactions are important. They take place, these are biochemical reactions because they take place inside the body, inside the organism's body structure. Now this copying creates 2 copies of DNA in a reproducing cell, and they will need to be separated from each other. Just keeping 1 copy of DNA in mother cell and pushing the other copy out does not work. So it has to create 2 copies of DNA, and it has to create 2 cells. One mother cell has to create 2 cells. Every DNA copy needs to have its own organized cellular structure for maintaining life process. DNA copying is accompanied by the creation of new cellular apparatus, and then the DNA copies separate. Every DNA copy has its own cellular apparatus. Thus, the cell divides into 2 cells. Okay. The simple example of reproduction in in amoeba, the parent cell, the nucleus divides. That means the DNA creates its own copy. So 2 DNA copies from one parent copy, and then the cell apparatus divides into 2. Okay, and then the division completes and 2 daughter cells are formed. So it has to be a complete division of the cell apparatus and copying of the DNA information that goes separately into each daughter cell. The copying reactions, that is biochemical reactions, they are not perfect in general. So the offspring, that is daughter cell, don't get exactly same copy as the parent cell. There is a variation expected every time a copying of DNA takes place. So there is a little bit variation every time there is a cell division and there is a copying of DNA. Now DNA copies generated are similar, but they may not be exactly identical to the original and exactly identical to each other. Sometimes these variations are so drastic that new DNA copy cannot work with the cellular apparatus it inherits. So they mismatch. In this case, the newborn cannot survive. But other variations are not that drastic and the offsprings usually survive. The surviving cells are not identical. They are similar to each other, but they are not exactly same to each other. This inbuilt tendency for variation during reproduction is the basis for evolution. Okay, and evolution is a separate chapter we are going to see on this channel. I'm going to take it for you. The importance of variation. Now, the place of an organism in the ecosystem and the design of the body, the place of the organism in the ecosystem is dependent on the design of the body of the organism, and it is also dependent on the ability to reproduce. So in the ecosystem, the place of the organism will depend on its design and its ability to reproduce. Okay, we'll see this little bit in more details now. The population of any organism has a specific position, that is niche, in ecosystem. So if you take this ecosystem here, so there are planktons, bacteria, worms, snails, frogs, these plants, fish, different organisms in this ecosystem. So the position is very specific. So population of any particular organism has a specific position in the ecosystem. This position is maintained due to ability of an organism to be able to make copies of their DNA during the process of reproduction. So ability to make the copies, that is ability to reproduce, is very important to maintain this position. Reproduction is therefore linked to the stability of the population of the species. Okay. An advantage of having variation within a population is that some individuals will be better adapted to their environment than the others. So say in human beings, if you take 100 human beings, some of them will be more fit in cold to survive in cold weather. Some of them will be better fit to survive in hot weather. Some of them will be more resilient, resistant to the viral infection like COVID infection. Some will be better protected against heart diseases. So the population, okay, has a variation. An advantage of having variation within population is that some individuals will be better adapted to the environment than others. Those individuals who are not well adapted to their environment are less likely to survive and reproduce. Those individuals that are best adapted are more likely to survive and reproduce, passing on their valuable traits, favorable traits, that is adaptations, to their offsprings. So say in a cold weather, particular population is fit, the few individuals in that population are fit to survive in that weather, in extremely cold weather. So these individuals that are fit, well adapted, will be able to reproduce. They'll be able to survive, then reproduce, so they can transmit their adaptations, that is favorable traits, to their children, to their offsprings. This process is called as natural selection, that is survival of the fittest. If you remember Darwin's theory from your previous classes, we will look into this in more detail when we go through the chapter on evolution. And this natural selection or survival of the fittest is important, is essential for evolution. Now, this variation, natural selection, is also important when environment conditions change. If there is a little variation within the population, the whole population may die out. That is called extinction. If there is a sufficient variation, some individuals will have adaptations that will help them to survive and reproduce. This will ensure their survival as a species. So the species will remain alive. It will stay and it will be able to produce offsprings and pass on their genes for the future, to the future generations. If they do not have variation, favorable variations, favorable adaptations, then they will not be able to survive and they will go extinct. Okay, so variation is important for the process of evolution, for natural selection and survival of the fittest. Now environment may change. So you may have a climate change, like temperature going up, water level going up, down, or meteorites may hit the earth. The introduction of new predator in that ecosystem might affect the population of those organisms. Changes in the prey, so their food may go up or down. Okay, so availability of different food sources may affect survival of the individuals of that population. And in those circumstances, the organisms that are best adapted will survive. They will reproduce and they will pass on those favorable traits to the next generation. There may be occurrence of diseases, the emergence of new disease or strain like COVID. So COVID did not affect cats and dogs as seriously as it affected human beings. So human beings were at risk. Okay, and in human beings also, there were certain individuals, certain races, they were at risk. So more people died from those races, from those countries. So population of bacteria living in temperate waters, and if the temperature goes up, certain population, certain bacteria will be able to sustain that high temperature and they can survive. But other bacteria, they may not tolerate high temperature of the water and they will die. So the variation is important for continuation of the species. Variation at individual level only lasts the lifespan of that individual. They don't help in adapting to environment over the generations. So if they say there is a variation in just a single individual, then it will not be transmitted as a variation of that species. Okay, because it will only last for the lifespan of that individual. It doesn't help to adapt to the environment over the next generations. Okay, variation at population level. So quite a few individuals at population level will help in evolution, sustenance and survival of the species against the change. Now this is explained in more details here. Say there are these animals produced. This is a deer farm. Okay, so there are excess animals produced, and if you see, if these excess animals have no variation and if the condition changes, then it will be difficult for this whole population to survive for long, to survive for many generations. Because if the weather changes, or if the predator changes, or such something changes, if diseases come, bacteria come, they all can die and they can become extinct. But the variation allows the individuals of the population to have different characteristics. So even if some calamity, natural calamity, artificial calamity comes, some of these individuals will be able to survive longer. They'll be able to reproduce and pass on the traits to the next generation, and these are called adaptations. The traits of those individuals that survive and reproduce will become more common in the population in the next generations. Okay, so you need variations, but not just at single individual level, but at the population level. Okay, so this is the initial part of this chapter. Now let's move on to the modes of reproduction used by single organisms. Now there is an activity here, activity 8.1, and we'll go through this activity. These are important activities, okay, remember, because activities give you practical knowledge and they make you understand the topic better. So what does the activity want? They want you to dissolve 10 grams of sugar in 100 ml of water. Take 20 ml of this sugar solution in a test tube and add a pinch of yeast. So yeast granules are added to this sugar solution. Put a cotton plug on the mouth of test tube and keep it in a warm place. After 1 to 2 hours, put a small drop of yeast culture from this test tube on a slide, cover it with cover slip and observe under the microscope. So in 1 to 2 hours, what would have happened? The yeast will grow. Okay, and what will you be able to see under the microscope? You will be able to see yeast, budding in the yeast. So yeast will reproduce because of the sugar solution. The parent yeast will give rise to different daughter yeast cells, and that's how the budding takes place. So single parent cell with the nucleus, formation of the bud. The nucleus also gives rise to the second nucleus. So the nucleus divides. This is a parent nucleus. That is a nucleus that enters into the bud. And there is a division that starts in the cell itself. The cytoplasm divides. The parent yeast cell stays intact, and this division completes, and there is a chain of these daughter yeast cells that forms, and you can see that under microscope. Okay, so that's your first activity. Now second activity is to take a wet slice of bread, keep it in a cool, moist, dark place. Observe the surface of slice with a magnifying glass regularly and record your observations for 1 week. Okay, so you can see this is a wet slice of bread, and what happens after a week? You will see growth of this mold here, bread mold. Okay, if you look under microscope or under magnifying glass, then you will see this fungus. This is fungus called rhizopus. Okay, so growth of a fungus. This is a fluffy and thread like. Okay, so there are threads and it looks fluffy. It is made up of interconnected network of filaments. These filaments are called as hyphae. H-A-P-H-Y-P-H-A-E. Hyphae. Okay. The hyphae together make up the body of the fungus. So this is the body of the fungus. Hyphae together, they make up body of the fungus, and it is called as mycelium. Okay. So remember, the body of the fungus is called mycelium. The hyphae are made up of cells, and these cells release enzymes that digest the material they are growing on. The digested material is then absorbed by cells. Most fungi are therefore called as saprophytes. So what does saprophyte mean? They grow on and decompose dead and decaying material. So they grow on dead and decaying material, and they also help in decomposing that material. So that is saprophyte. So fungus is a saprophyte, and the fungus bread mold is called as rhizophus. Just to revise, the interconnected network of filament is called hyphae, and the mass, the body of the fungus together, is called mycelium. Okay, and we are going to look at more details of the reproduction later on in this chapter. The yeasts are single cell forms. They reproduce by budding. Okay, so yeast reproduces by budding. Okay, and molds, bread molds or rhizopus, they are multicellular hyphae. In moist places, they reproduce by spore formation. So these are multicellular. Yeast is a single cellular, whereas rhizopus, mold are multicellular, and they reproduce by spore formation. So there's a difference between reproduction in yeast and in mold. Understand this. So the modes by which various organisms reproduce depend on their body design. So there are different modes of reproduction here. We'll go through them. These are described in your CBSE book. The first one is fission. For unicellular organisms, cell division or fission leads to creation of new individual. It's a simple mode of reproduction. Binary fission in any random plane. Okay, it takes place in amoeba. Binary fission in definitive plane takes place in leishmania. Leishmania causes kala azhar, black fever. Okay, multiple fission, they take place in malarial parasite, that is plasmodium. Now I will take you through these different fission techniques, the fission methods that take place in next few minutes. There is an activity here that wants you to observe a permanent slide of amoeba under microscope. Observe another permanent slide of amoeba showing binary fission, and compare the observation of both the slides. So amoeba is a single cellular structure, we know that. And when you see the binary fission, there is a division of this cell in a random plane. There is no particular plane, there is no particular line for division. It just takes place randomly. So it can divide in this plane or in this plane or in this plane, and it will ultimately give rise to two daughter cells. So it is in a random plane. So it is a binary, that means it divides into two. Bi is 2. So binary fission in a random plane. Okay. Now, next one is a binary fission in a definitive plane, such as in Leishmania. So Leishmania has got a tail. Okay. So if it divides in a random plane like that, then it will not create exactly similar type of cells. The daughter cells will not be similar to the parent cell, because it's not the round structure like amoeba. Amoeba cell division can take place randomly across any plane, and it will divide into two cells because it's rounded. But Leishmania is elongated, it has got a hair at one end, so the division takes place in a particular plane. It is not a random plane, it is a definitive plane. So this is a plane of division. Okay, it takes place in this plane. So one Leishmania cell gets divided into two Leishmania cells, the nucleus divides, the cytoplasm divides, and the cell division takes place, and one maternal cell gives rise to two daughter cells. So that is binary fission in definitive plane. And the third type of fission is, it is not a binary fission, it is a multiple fission. So if you see malaria parasite, that is Plasmodium, this is a parent cell, it is called sysont. It has a nucleus, cytoplasm. Now it doesn't divide into two, it divides into many daughter cells, that are produced by multiple fission processes. So multiple fission gives rise to multiple daughter cells that are released when the mother cell bursts open in the blood. Okay, so that is about fission. The second mode of reproduction in the simple organisms is fragmentation. Okay, now fragmentation is breaking up, right? So there's a activity that your book wants you to do. They want you to collect the water from a lake or pond that appears dark green and that contains filamentous structures. So put one of the filaments on a slide, drop a glycerine on this filament and cover it with cover slip and observe under microscope. And they want you to look at the filaments that are Spirogyra filaments under the microscope and how they look. So Spirogyra has got a simple body. It's got cells, it's a multicellular, but it has got a simple body. Okay, understand this. Spirogyra cell. So it has a simple reproductive method as well. It simply breaks into the pieces, smaller pieces. When it matures, it breaks into smaller pieces. And these pieces or fragments, they grow into the new individual. Okay. So mature Spirogyra organism breaks into different pieces and each piece grows into the different new Spirogyra organism. So this is a simple multicellular organism, but this cannot take place in complex multicellular organisms like human beings. Can this type of reproduction take place in human being? If you break down human being into 10 pieces, can the pieces grow into different human beings? No, because it's a complex multicellular organism, human being. What happens during the process of fragmentation? A fragment of a parent forms a new being. It occurs when a shoot that is rooted becomes detached from the main group. So different filaments are there and they break up into pieces, and each piece gives rise to a new organism. Multicellular organisms with simple body organization can divide or reproduce by fragmentation, but it cannot be used by all multicellular organisms with complex structures cannot undergo cell by cell division. This is because in complex multicellular organisms, each set of cells have specialized function. They are called tissues. So cells give rise to tissues. Tissues form the specialized tissues form the organs, and different organs, they are the part of the whole body. So that's why one cell cannot give rise to a specialized tissue of other type. It will only generate that specific cell type. That is why complex multicellular organisms cannot reproduce on the basis of cell by cell division, like it can take place with Spirogyra, we have just seen. Okay, so these specialized cells of the complex multicellular organism are organized as tissues, and they are placed in organs. Organs are placed in different positions in the body. So fragmentation doesn't work. It is a type of asexual reproduction in which an organism simply breaks into individual pieces at maturity. And these individual small pieces then grow to form new organism, that is in Spirogyra. That's the summary of fragmentation. And again, to revise, cell by cell division is possible in unicellular organism like amoeba, in simple multicellular organism like Spirogyra, but multicellular complex designs, cell by cell division is impractical. Why? Because different cell types perform different specialized functions. Reproduction is also the function of specific cell types. A specialized reproductive cell is capable of growing, proliferating, and making other cell types under the right conditions. So reproduction is a function of specialized reproductive cells. These grow, proliferate, multiply, and they make other cell types under specific conditions. Okay, so that is about the fragmentation. Now third way of reproducing is regeneration. Now ability of a fully differentiated organism, that is mature organism, to give rise to new individual organism from their body parts. Okay, so if you cut one body part of that organism, that body part will again develop into a fully differentiated organism. This is called regeneration. If the individual is cut or broken up into pieces, these pieces will grow into separate individuals. An example is Hydra and Planaria. So let's look into this to some extent. You don't need great details at this level. Okay, so these animals can be cut into different number of pieces, and each piece grows into a complete organism. This process is called regeneration. Hydra possesses tremendous regeneration potential, and it is capable of regenerating complete organism from small tissue fragments or even from dissociated cells. So not just the piece of Hydra body, but even a small piece of tissue or even from the separated cells, it can give rise to a different new organism in Hydra. The other examples are planarium and starfish. Okay, regeneration is carried out by specialized cells. These cells proliferate and make large numbers of cells. From this mass of cells, different cells undergo changes to become various cell types and tissues. So I'll explain you this in this diagram. So say we cut this hydra into two pieces. It's called amputation. Amputation is cutting the body part. Now these two parts will regenerate. Okay, so what happens is there are specialized cells in the body of the hydra. These specialized cells, they proliferate, that means they multiply, and they give rise to a large number of cells. The mass of these cells, okay, that undergoes changes, and that gives rise to various types of cells and tissues, okay. And then these changes take place in organized sequence in a process called development. So the whole process after amputation is called development, okay. So development, and understand again, because this is slightly complex, the specialized cells in the body of the pieces of the hydra, they proliferate, they make large number of cells, that creates a mass of cells, and then from this mass different cells will undergo changes, and they will give rise to different cells and tissues, and that will cause regeneration of the remaining part of the body. So here the remaining part is this upper one, here the remaining part is the lower one. It will be regenerated by these specialized cells in the hydra, and the whole process is called development, okay. So regeneration is not exactly a reproduction. It's not exactly same as reproduction, okay. Understand. The next method of reproduction is budding, and again the example is hydra. We have just seen the regeneration takes place in hydra, and I've explained you this example. But regeneration, I gave you examples of starfish and planarian. Again, budding, the example is hydra. So this is the body of hydra. A small bird arises from the body of the hydra, and then bird develops into whole new hydra, and then it grows and matures. So hydra-like organisms, they use regenerative cells for reproduction in the process of budding. So remember this word. So this is a regenerative cells to develop a bird, and that bird grows into a new hydra. In hydra, bird develops as an outgrowth due to repeated cell division at one specific point. So regenerative cells will proliferate, and that bird will develop into new tiny individuals. When fully mature, they detach. So when it matures, it detaches from the parent body and now becomes a new independent hydra, okay. So that is budding. Now next is vegetative propagation. So it's very important, practically relevant point here. So what is vegetative propagation? So plant parts like root, stem, leaves, they develop into new plants under appropriate conditions, okay. So the new plant is developed from root, like in banana, stem, like in sugarcane, or new plant can also develop from the leaves, and the example of that is leaf of bryophyllum, okay. So I'll explain you again the leaf of bryophyllum. So this does not take place, the vegetative propagation does not take place in most animals, but some plants can use this mode for reproduction. It is used in methods such as layering or grafting to grow many plants like sugarcane, roses, grapes for agricultural purposes, okay. So if you look at sugarcane, how sugarcane farming is done. So pieces of sugarcane stem are buried into the soil, and from the birds, so there is a bird at the nodal point, and this is the internode, bird at the nodal point, that bird grows into a new plant, okay. And that's how sugarcane farming is done. So this is a vegetative propagation, that a stem gives rise to the new plant. Another example is a grafting. So there are two different varieties of the plant, the stems of two different plants are connected and they are kept together, wrapped in such a way as shown in this diagram, and then they just behave like a single plant, and the plant grows and develops into a new plant. So this is a vegetative propagation from stem again. Now, what are the advantages of this vegetative propagation? So plants can bear flowers and fruits earlier than those produced from the seeds. So it increases, it makes the process of flowering and fruit bearing faster than it will be with the plants that are produced from the seeds. It also makes it possible to generate new plants for banana, orange, rose, jasmine that have lost the capacity to produce seeds. Okay, so the plants that do not produce seeds can be regenerated, can be produced with the help of vegetative propagation. So the first advantage is it makes it possible to bear flowers and fruits earlier, so time saving. It makes it possible to propagate the plants such as banana, orange, rose, jasmine that have no ability to produce seeds. The third advantage is all plants that are produced by vegetative propagation are genetically similar. So the parent plant, its characteristics can be maintained in the baby plants or daughter plants. So if you have good variety of sugarcane, good variety of banana, you can expect that the baby plants will have good banana and they will be good product as well. So that is the advantage of vegetative propagation. There is a small experiment your book wants you to do to demonstrate the example of vegetative propagation, and that's with the potato. Take a potato and observe its surface. It will show you notches, okay, on the potato. Cut the potato into small pieces such that some pieces contain notch or bud and some don't. Spread some cotton on the tray and weight it. Place the potato pieces on this cotton. Note where the pieces with the birds are placed. Observe the changes taking place in these potato pieces over the next few days. Make sure that the cotton is kept moist. Which are the potato pieces that give rise to fresh green shoots and roots? So it is obvious the potatoes that have notches, the pieces that have notches will give rise to the fresh green shoots and roots. So you can see I haven't got the diagram to show you the pieces, but if you just leave the potato as it is in a moist condition, then the places where there are notches will give rise to shoot and root. Okay, shoot and root. So that's the easy experiment to do. Now similarly, so these birds can be produced in the notches along the leaf margin. Okay, so the notches on the potato pieces, notches along the leaf margin, they can give rise to birds, and these birds can develop into new plants. So when this leaf falls on the soil, it will give rise to the birds at its notches and they will develop into the plants. So example is bryophyllum. Okay, bryophyllum, remember that. And there's another activity the book wants you to do, very interesting activity. You must all have seen money plant. So they want you to take money plant pieces in such a way that they contain at least one leaf. Okay, why? Because that leaf will carry on photosynthesis and it will produce food. So cut out some other portions between two leaves, that means just stems, okay, and the portions with leaves. So there are two types. Dip one end of all the pieces in the water and observe for a few days. Which one will grow? So the observation will be money plant stems with leaves will grow, and without leaves will not grow. And what is the conclusion from this observation? So because the leaves can produce food by photosynthesis, this portion, this piece of the money plant will grow, and because there is no leaf to do photosynthesis here, that will not grow. Now let's see more details of this. So money plant is a perennial plant, that means it grows all the year, around the year, okay, perennial, and it grows in Asian region. We call it a money plant because the leaves, they look like coins, okay, they're round, coin like. The money plant can also reproduce through vegetative propagation. When we cut this plant into parts, it survives by doing photosynthesis and creates another money plant by vegetative propagation. But the cut part, if that does not contain leaf, the plant is unable to photosynthesize and produce its own food, so it becomes yellow and then eventually it dies. Okay, so this is the activity of money plant that also grows by vegetative propagation. Okay, so remember, vegetative propagation is seen in many plants, the household plants, indoor plants, as well as it is used in the agricultural field to plant various to do various crops. Now there is a activity, in fact it's the information rather than activity. Your book gives little bit information about tissue culture, which is very prevalent in modern day agricultural industry. So what happens is, in tissue culture, I'll show you this diagram to explain about tissue culture. Okay, very simple diagram. New plants are grown by removing tissue or separating cells from the growing tip of the plant. So say if this is a plant, so growing tip of the plant, wherever the tips are, the tissues and cells are separated. Okay, and the cells are then placed in an artificial medium where they divide rapidly to form a small group of cells called callus. So these cells are kept in a medium. So what is medium? The medium is a material with favorable nutrients in that. So these cells will grow, and they will grow to give rise to a group of cells called callus. Okay, callus, callus. Now callus is transferred to another medium containing hormones for growth and differentiation. So they have, once the callus is generated, it is put in another medium, and what is there in the medium? There are nutrients and hormones for growth and differentiation. Okay, so hormones for growth and differentiation in this medium, the callus is added to that, and then there will be a formation of small plantlets from the callus. So you can see that small plantlets. Okay, and these plantlets are then removed from this medium and they are planted into the soil, and then these plantlets, when they grow up a little bit, they go to the farms, and then the farmers, they plant these into in their farm, and that's how the tissue culture technology is used in modern day farming. So what is the advantage of this? Using tissue culture, many plants can be grown from one parent in a controlled, disease free condition. Okay, so you can do this in laboratory, you can do this in an industrial setting, and that's why you can produce lots of plants from one single plant. And this Environment here is disease free, infection free. So the large number of plantlets will survive. So this helps to increase the number of plantlets, and it is used commonly for ornamental plants. Okay, so this is about tissue culture. Now the last part of this this section of the reproduction is spore formation. Okay, now spore formation is asexual method of reproduction used in rhizopus. You remember the braid mold has got hyphae, a network of filaments called hyphae, single hypha, plural hyphae. Okay, now this network is non-reproductive, but there are tiny bob on stick structures. So bob on stick structures, they are reproductive. They are specialized tissue, specialized cells. They are important in reproduction. So hyphae, that is a network of filaments, is non-reproductive, but bob on stick like structures are for reproduction. The blobs are sporangia, so single sporangium, multiple sporangia. Okay, remember this, single sporangium in this diagram, single, and multiple sporangia. Okay, so these contain cells, spores, that can develop into new rhizopus individual. So these spores, these are cells, when they burst, they come out when the sporangium bursts, and when the spores get good moist environment, moist surfaces, then they can develop into new rhizopus individual. So spores have got thick protective cover for their protection, but when they come in contact with moist surface, they grow into new rhizopus individual. Okay, so to conclude, asexual reproduction, the mode of reproduction that allows new generations to be created from a single individual. So you do not need two individuals of that species. Single individual gives rise to the offsprings, that is new generation. All the above modes that we have discussed in this video are asexual reproduction modes. Okay, because there is a generation of next generation from a single parent or mother cell. That's why they are asexual reproduction methods. Now in next video we'll see sexual reproduction methods. So I'll see you soon in the next video. Meanwhile, you go through this video and I'll see you soon. Take care. Bye bye.